Yeah, so Angelika, so we are so happy to have you here in this conversation, and we heard you also prepared a slide for us, correct?
I did.
I mean, your project, I mean, it was so much fun to read, and we already know you also for some time, and you gave a great presentation about your research a couple of years ago at our user meeting. And we also have some good news for you. And so actually, before you give your presentation, we wanted to let you know you are the winner of the SyncroPatch Grant 2020.
Are you serious?
Yeah, totally serious.
Is that true?
Yes, congratulations, Angelika.
Das ist ja super. Thank you very much.
Yes,
it's really great.
Yeah, it's true.
Wow.
So we wanted to take out the stress before you present.
That's very nice. Well, actually, I did prepare. And the whole company was saying, "Hey, go for it. We need this thing."
yeah, you still have to present.
Sure, I will. Yeah. This is great. Yeah. Wow. Yeah. Thank you so much.
You know, we were the 3 judges, and from all 3 of us, you were in the top 3. So, you know, we independently were judging. And then we pulled all the results together. And so, yeah, you are the winner.
May I ask how many applications you get for that?
More than 40.
Wow. Wow.
Yeah, so we had a lot of stuff to read, I tell you. But it was great fun also.
So it was such a difficult decision. We decided we will give one to a team in the US and one to a team in Europe. Yeah, they were such great quality.
Yeah, so what is going to be your first experiment?
So our first experiments are actually already running because we are putting together the stable cell lines of certain NAF mutations. And we are, of course, we wanted to have them for manual patch clamp, but now we can put them on the SyncroPatch, which is super cool. We still struggle to express NAV 1.8, but we have a new system and that may be helpful. Yeah, and so we've got these 10 mutations that I mentioned in the grant application already, which we think are really interesting, especially because we also have the iPS-derived cell lines there. And so we would like to start with them, get used to the SyncroPatch, and then as soon as we've got it running, then, I mean, now we can buy the library, the pharmacological library, which is great.
Yes, fantastic.
Yeah, because I mean, obviously we were waiting. We were thinking that this would be cool, that we could screen these the drugs, but we didn't have the devices and screen them on all sides. I don't know whether this is really, yeah, right. And now we can do it on the SyncroPatch, and so we buy the library. And then as soon as we've got a biophysical, the protocols up for sodium channels, and I think you already have some protocols there,
yeah.
Then maybe we would like to tweak it a little bit so that we can also do research in currents, and also we would like to check for temperature changes as well. Mm-hmm. This is really important for the pain patients, as you can trigger with temperature change the symptoms. Yeah, and then we will screen the FDA-approved drugs, which we would like to look at. And yeah, and when this is running in parallel, what we would like to do is try to get not only rodent DRGs, but also pig DRGs in a preparation in which we then can do the screening on the DRGs in the SyncroPatch. Yeah, and then the third part, which I think is the most interesting, but also the most challenging, that would be to finally get the iPS-derived sensory neurons screenable, to get them on the SyncroPatch. And this is the part where now I can be super honest. Yeah. I would have been honest as well before, because there I cannot promise that it will work. We would like to get it work, but you know how complicated these cells are, and so it's possible that we cannot achieve it. But I think we just need to try it. And in the moment we are optimizing the protocol, and we've got really good successes there. So I think, I mean, we can fax the cells. So why shouldn't we be able to put them on the SyncroPatch?
Yeah. Yeah. No, it sounds perfect. And this was also one of the reasons what we liked so much about your project, that you had, you know, the stable cell lines, which will 100% be sure to be able to run on the systems. Then you have the DRG neurons, which will be a bit more difficult, I think, but for sure doable. And then you have the third level, which is a really high risk, but that's okay because you have this nice mixture of, you know, low risk, intermediate risk, high risk. And I mean, we are happy to help you in all the stages. And we would also love to make them work, right? I mean, it would be also fantastic for us.
Yeah, yeah. And I mean, we already have this idea with Irina Vetter, where we would like to check the toxins. So, I mean, that works very well there. Yeah. Yeah, but I think what is really, really nice in Aachen at the moment is that we're having this dynamic group of sodium channel researchers, which has recently formed. And it's really, maybe I show the slides, you know?
Yes.
Yeah. Okay. So yeah, so in Aachen we have the Sodium Channel Network Aachen, SCNA. We needed to take this name, right?
Yes.
How long did it take you to come up with that?
Well, actually, there were some discussions involved because, I mean, yeah, it's maybe a bit too obvious, but yeah, I like it.
It's fantastic.
And it's about the sodium channel related pain disorders, where we are looking at the molecular mechanisms, and the goal is personalized treatment. And these are the people involved. So it's basically electrophysiologists, clinicians, geneticists, and computer models. And this is the topic, we talked about this. So these are the electrophysiologists. Of course, my lab, but also the lab of Günther Schmalzing and Ralf Hausmann. They are in the pharmacology, and they have really good experience with this drug screening, and also single channel recordings and biochemistry. And the biology department is involved with Marc Speer and Markus Rothermel, and then Jan-Philipp Martens, who used to be with Christoph Falke in Jülich. And he is really in-depth electrophysiologist. And then we've got the clinicians who have this large cohort of pain patients. 200 patients are already here and ongoing. They do in-depth phenotyping with QST, quantitative sensory testing, fMRI, EEG recordings, and we do stimulations on the fMRI. And so we get highly phenotyped patients, which then we do, of course, the genetic screening together with a human genetics department, Ingo Kurt, who has already a lot of experience with sodium channels and neuropathies. And then we put everything together, and we are working on computer models, which are now possible because we've got these really good computer structures, these cryo-EM structures of the sodium channel. And Giulia Rossetti and Jan-Philipp Martens are really good in molecular dynamics and in silico drug development. But obviously, we just had in silico drug development, and we didn't really have a potential or a possibility to do this this screening on a high throughput thing. And so the SyncroPatch is super here. In addition to this network, we also have a consortium together with Grünenthal, and this is called Bio2Treat. And there, Grünenthal is developing a pain watch, which we would like to give the patients so that they can track all the biometric input and informations of their everyday life that may trigger their pain attacks. And then we take iPS cells from these patients and differentiate them. And then we would like to integrate all that information in order to find out what triggers individually the attacks. Yeah, and Grünenthal, with them, we also work quite closely together with the iPS cells. And they are really interested in that machine as well. They don't have one, I think you know. But of course, it would help as well if we get it going. Yeah, and then, so what would we do? This I already talked a bit about, the electrophysiology basics, biophysics at first with the mutations. Then we would do the drug screening, and then in the end, we would like to do the translation with the hit validation and in the different species, like in the mouse, pig, or then in the end in the humans. Yeah, so these were my 2 slides.
Yeah, perfect. I mean, you also already covered some of the other questions I had. And is there something especially why, you know, the SyncroPatch fits so well for your lab?
Yeah, so what I really like about the SyncroPatch is that it makes really high quality recordings. So you get the giga ohm, and that's for sodium channel recordings really important. I mean, yeah, I'm preaching to the choir here. So that's yeah, no, this is really great. And then you can change the temperature, and I think you can.
Yeah, so it's I think it's a fantastic fit, and I mean, I can see that you would love to start tomorrow and already preparing it for it. It will be so much fun, I think, when we are working on this project together. Then, do you have already some plans, maybe for the time after the 9 months?
Yeah, so we've got two approaches. So one would be that we would integrate it in the EZKF core facility. So we've got a core facility which provides fax, and then also what do we have, immunostaining and mouse facilities. And so we were thinking of trying to implement it as an electrophysiology core facility. This would be one approach. The other approach would be that we would write a Großgeräteantrag. We've got about 12, 14 persons being interested already at the RWTH. And so I think we have enough impact in order to get a large enough group together in order to bring that to the DFG and then try to get it through. Yeah.
Perfect. I mean, even two approaches. I mean, that that's fantastic. As I hear you, I'm more and more convinced we did the right decision. Yeah. It's really difficult between all these excellent applications, but I mean, yeah, no, this is a great project. And
yeah, it took a long time to finally make me a Nanion user, a real one, right?
Yeah, a real one. Yeah, I was just saying to Ellie that I remember when you came, I don't know, maybe 8 years ago to Munich, that when we measured the resurgent current together on the patch liner.
That's true.
Which was already a lot of fun.
That was right, yeah.
Now we're going to measure the resurgent current then on the SyncroPatch.
In big scale, right?
In big scale, yeah.
Yeah.
I had a quick question. How large is this library that you were thinking of purchasing, and if you can describe it a bit?
Yeah, so it's called, I need to read, I'm sorry, it's called Spectrum Collection from Microsource Discovery Systems. And it's a collection of FDA and EU approved drugs, which are currently on the market. And so the idea would be that we screen all of them. It's 1,760 drugs. And it's all of those being approved. And and the idea would be that we could find something that then we can use off-label, and like this we would avoid the long approval thing, which then in the end may not help the patient anymore. And then you could additionally buy 800 biologically active substances. They are not in these in this first pack, which then could also maybe be screened. But we would like to start out first with the FDA-approved drugs.
And then you mentioned toxins. I think at some later point you were thinking of doing.
Yeah, so the toxins, of course, are not for treatment in the end. They are more for biophysics and interest. And this is a project that we currently have together with you guys and Irina Feta in Australia. And there we wanted to see whether we can target single sodium channels like NAV 1.9 with specific toxins in order to knock them out and get a better mechanistic feeling for what they are doing in the iPS-derived neurons or whatever neurons. Yeah.
Wonderful. Thank you.
You're welcome.
Yeah, it's I'm so happy also.
Oh, me too. Yeah. So you guys are all at home as well, right? So you're corona affected, right? Yeah.
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 So yeah, so you can see all our different homes.
Yeah. I had to give lectures from home, so I try to keep it tidy. But you'll see my my sodium channel here.
Oh yeah.
Can you put that around your neck? Is it like a
no, well, that's the sodium ion pathway through the sodium channel. It's too large. And I'm a nerd, but not that bad.
Earrings.
No way.
We can just congratulate you again.
So much. Yeah, I will make some phone calls now.
Yeah.
We're so pleased that you put in your application and that we decided that you're the winner, because as you say, it's about time that you're a real Nanion user.
It's so hard.
Official.
Thank you so much. I'm really, really happy. I really appreciate it. I'm really glad that we could convince you. Cool.
Yeah. You could open the bottle of champagne and enjoy the evening.
Yes, alone in front of a Zoom screen. So enjoy the evening as well. Thank you so much.
Thank you. So,
congratulations.
Thank you. See you soon. Bye-bye.
Yeah,
Bye.
bye.
Bye.